Yes, hello. My name is Bas Voermans. I'm joining as a PhD student of the Amsterdam University Medical Centers on the location University of Amsterdam, where I'm very happy to present our paper, which is titled Microbe2Pixel Taxonomy Informed Deep Learning Models and Explanations. It's a bit of a mouthful, and if I show you the schematic of everything we have done in this paper, it might be looking a little bit daunting and a little bit complicated, but hopefully at the end of my talk, this will all become a lot more clear. Um, Firstly, what do we do as a group? I'm part of the bioinformatics department, where we really specialize in using data of the human gut microbiome, which we then relate to health and disease based on the microbes that we find in the gut. And typically, this can be done in many different ways. But what we specialize in is the use of machine learning in this regard, where we can use the microbiome data, which is very feature rich. So we have many, many times the amount of features compared to the amount of samples. Where we use machine learning not really to develop a machine learning model to be deployed in the clinic, but rather we are interested in the mechanism, the interaction between the microbiome and the human phenotype, let's say it that way. So that's a little bit of our background. And yeah, we do this research with the use of machine learning. And yeah, typically what we use are these very well established ensemble models like XGBoost, CutBoost, or extremely randomized trees, randomized forests, you name it. And these models are really developed as tabular data models. So we tackle the data as a tabular data problem, where you can think of the data somewhat as a spreadsheet, where each microbe is seen as a feature, and on the vertical axis you would have your samples, and you would see here that this specific Prevotella species has been read 860 times in sample 1 and 0 times in sample 2, yada, yada, yada. And what would happen if you do this, train one model on dataset A or train a model on dataset B, is that you would start all over again. The model is completely naive in what it is looking at. However, we as humans or as researchers and the literature, the corpus of literature on this topic is not naive in this regard, because we know a lot about these microbial species from previous research. For instance, what we can always know is the taxonomic tree of these microbes, or the tree of life, as you can call it, where every organism on Earth fits somewhere in this tree of life. So if we take two random organisms from Earth, we can always find a distance or a similarity between these two organisms in this taxonomic tree. And what we sought out in this research is to use this taxonomic tree to kind of help our machine learning model out a little bit, to urge it in a direction that some microbes are more similar to each other than others. And this might also account to their function a little bit. So what we use actually is taxonomy as a proxy for similarity. And from this taxonomic tree and the microbes we find in our data set, we can get a distance matrix of microbes, which is basically features by features large, similar to a correlation matrix, or a distance matrix like that. And we can then use a dimensionality reduction like multidimensional scaling. And we have used multidimensional scaling, where we transform this distance matrix into coordinates. And if we would take two-dimensional coordinates, so we would do multidimensional scaling to get the first two principal coordinates, we can actually get locations on an image or locations on a map, however you would like to call it, which allows us to actually place each specific microbe somewhere in an image. So what we would get is one set of locations for our data set, which is conserved for every sample. So in every sample is represented as an image, and the locations of the microbes in the image are conserved. The only thing that changes is the pixel intensity and this changes with the relative abundance of each of the specific microbes in that sample. And this is just a highlight to show what it kind of looks like on a widescreen. Actually, when the intensity is higher, the pixel becomes more white, so it's actually the inverse of what I showed you. So it will look something like this. And this is an actual example of the dataset that we used and the locations we found from it using that taxonomic tree. And what you can clearly see is that there are clusters forming. For instance, I don't know if you can see my mouse, but on the right middle part of this image, you can clearly see quite a large cluster of microbes forming, which is then part of a larger cluster of clusters, basically something like that. And we hypothesize that these are because we have chosen our distance matrix to be dependent on the taxonomy. And as you can clearly see here is that if we color them by phylum, which is a very high level in the taxonomic tree, we color the top 5 phyla. And you can see clearly that the phyla are clustered as larger clusters of subclusters of different taxonomic ranks, basically. So every taxonomic rank is clustered by itself and is then also part of a bigger cluster of a higher order in the taxonomic tree. And you might be wondering, why are we actually using these images? Why go through all this trouble? The tabular methods work fine. Why not do some deep learning on tabular data? Well, you might be a little bit true that those methods works perfectly fine, but this use of images also allows us to use the modeling complexity, which has more successfully been applied to images as it has been for tabular data. So what we can now use is the concept of transfer learning. So training a model, using a model that has been trained on a ginormous data set like ImageNet, and applying it to our microbial images or microbial pathology slides, so however you would like to call it. And the way this works is that we use this pre-trained model, we rip out the last layer, and we plug in a new layer, and actually we can only train the last layer as an output or retrain the entire model, and this is something you can choose depending on your data problem. And the concept to explain how this actually works is can be quite easily done with this tiger example, that in the first layers of a neural network, a model actually learns things like edge detection and shape detection and more basic things which are relevant regardless of what you are looking at. And then the further you pass down into the model, it's generally hypothesized that the model then combines these shapes into larger substructures, which are then finally built into a real representation of what this model is looking at. But what you can imagine is indeed that these two first layers, they're not only relevant to looking at tigers and distinguishing them from lions, but they can be applied to all sorts of fields, because these concepts of looking at shapes are important whatever you are looking at. And you might remember that I started my talk that we are not really in the business of deploying these models, but more in explaining the microbial data with the models. And the use of images also allows us to use the more sophisticated explanation methods which have been developed for images. One of those is called LIME, which stands for local interpretable model agnostic explanations. So local means that we can explain on a sample level, which is very important in the clinic, where you would want to see which microbes are important in sample A, which microbes are important in sample B, and whether this changes Not only that, which is important across this entire data set, because it can be very sample driven. Interpretable means that it's very easy to understand. Also for clinicians, this is very important. They typically don't have a lot of machine learning experience. Model agnostic also means that it is possible to be applied to many different models. So we have now chosen a specific efficient net architecture, which has been shown to work very well on ImageNet without additional data. But in the future, there might be way better models, something like image transformers, which this method can easily be interpreted or applied to because it is model agnostic. It doesn't really care which model is behind the predictions. So how do we actually use Lime in our model? I will go over it briefly, how Lime works. It's basically that you take this original image, which is the first day that I actually got my access card to the Amsterdam University Hospital. So you take this original image and you use some sort of segmentation algorithm to segment the image into different superpixels. And what you then do is you get a number of perturbed images in which some superpixels are turned on, some superpixels are turned off. And for each of these perturbed images, you get a prediction from your model. So you infer your model many, many times. And you also calculate the distance between each perturbed image and the original. So some superpixels might be bigger. And in that case, the distance between the original image is, of course, bigger. And what you then do is actually fit a weak learner, something like a linear model, which is very easily interpretable, much more interpretable than a deep learning model like EfficientNet. You fit it to the perturbation matrix, basically, and you will get an importance measure for the importance of each superpixel in your image according to your model, basically. And this can be indeed done on a local scale. And generally, it is regarded that permutation feature importance, something like that, is a very good factor for feature importance, very robust. However, it can only be done really on a global level. And you can never really go from global to local. You can only go from local to global by the use of aggregation. So you might be wondering, what is this added benefit of local? Why go through all this trouble? Which can be quite easily explained with a very well-known example of the very often used Titanic benchmark dataset, which is, I think, the default on Kaggle still. If you log on to Kaggle, it will really ask you to start off at Titanic. So any machine learning researcher starting off will probably come across this dataset at some point. If we use SHAP on a global level to explain which passengers are likely to survive the disaster of the Titanic, you can clearly see that sex is very important. And this actually makes sense because from history we know that it really was women and children first on the life rafts, and after that the men could take place if there was space left, but it generally was not really the case. Deck is also really important, which is interpreted as if the deck you were on had life rafts, the chance of survival becomes markedly greater. But this is only really on a global level, but if you look on a local level, and we then look at the first few classes of the passengers, we can actually look at the interaction between the sex of the passenger or the gender of the passenger and the class they were in. And what you can then see is that in the first two classes, you can clearly see that the distribution of importance of the female gender is a lot higher and is regarded in SHAP values as having a larger chance of survival than the male gender. But however, in the third class, you can really see that actually the model thinks that males have a much higher chance of survival, and this can probably be attributed, or we can hypothesize that indeed women and children first was really only the case in the first two classes during the sinking of the Titanic, and that if you were in third class, regardless of being male or female, you will not be able to step foot on a life raft. So everybody in third class really went into the water, and at that point, males have a little bit higher chance of survival because maybe some physical attributes of males might be beneficial for chance of getting making it until the lifeboats arrive. So this is why we actually chose local, because it allows us to look at more closely into these interactions, why we are really interested into developing a local method for microbiome data as well. So we used a publicly available cirrhosis data set published in 2014 by Qin et al., a Chinese paper, which had shotgun fecal metagenomic sequencing of about 130 patients. And what you can see is that they had 10 times as many features as instances in the data set, where approximately half of the data set was healthy control participants, and the other half was people with liver cirrhosis, which was mostly caused by alcoholism, but some were also non-alcoholic cirrhosis. And the objective of our machine learning algorithm in this case is to classify healthy versus disease patients based on their microbiome sequencing results. We benchmarked our approach compared to extremely randomized trees, which is an implication of random forest, extreme gradient boosting, which is very well known in the machine learning world, and also, deep learning models using our Microbe2Pixel algorithm to get locations, but also getting an image of random locations to see what actually the influence of using the true taxonomy is compared to using images without taxonomy. And this is the results we came up with at the end. What you can see is that taxonomy really improved our results quite a lot, actually. However, if the goal of your model is to look for out-and-out performance in machine learning, then the established models are still the best. But as I said, we are not really interested in deploying our model, where we're looking at explanations to link the microbiome back to the phenotype of a patient. And in that case, we are looking at feature importances. So the true result of our research was not really in the modeling performance, because we had really expected that the model performance would probably not stack up to the state of the art in machine learning for tabular data. But we were hoping to see some results in the feature importances instead. So this is an example of the explanations that you will get. And because we use taxonomy, the clustering of importances is also based on taxonomy, or is more driven by taxonomy than in the other cases. And these were the top markers from each of the feature importance algorithms that we used. And as most of you probably are not microbiologists, I don't think it's very necessary or indeed very interesting to go into whether these markers are actually a better representation of what might be driving liver cirrhosis in these Chinese patients. Rather, we chose a more quantitative approach to testing feature importance algorithms or comparing them to each other. So what we did was we first tested our model using the entire trainings data set, basically. And we did this multiple times. But also, we trained our model using only 50% of the training samples. And we compared, we did this sampling 50% 10 times for each stability run we did. And what the hypothesis here is that the robustness can be measured by using only 50% of the training samples. And the closer you get to the feature importance set with all training samples, the more robust your feature importance algorithm becomes, and thus the feature importances you get from that algorithm will generalize a lot better to instances where you might not have a lot of training samples. So you can approach your final feature importances with unlimited samples a lot quicker with a method that has lower difference when you only use 50%. And what we can then see, as can be read in our very nice paper, that Microbe2Pixel actually gets a lot lower sum of squared errors in the feature importances, both on local explanations as well as global explanations. So it doesn't really matter which of the two you prefer. Our method, if you have little training data, which is almost always the case in the microbial field, this approach really allows you to have more robust feature importances out of your data. Typically, when we link the microbiome and these stop markers back to the phenotype, we not only look at, we don't really look at actually all the features. We are not looking at 1,300 features every data set, basically, but we are rather looking at the top, let's say, 25, maybe 10 to 200 markers maximum. And here you can see that actually for both local and global, again, on the top markers, Microbe2Pixel has a lower sum of squared error when you use only 50% of the training data. So you approach the same set of importances. Thus, Microbe2Pixel importances are a lot more robust than the other methods. However, you can, of course, be robustly wrong, and here it might become a little bit more biological. But for cirrhosis, it is very well known in literature that there is an association between oral microbes ending up in the gut when liver cirrhosis is present in a patient. So it didn't really come as a surprise that these species or genera of microbes came up to be important in this data set as well. So what we really know is that, yeah, the microbes in actually the stool become more like the microbes in the mouth in cirrhosis, and we can clearly see that these top genera, or you guys probably will not know, but these are really very well-known oral genera. And yeah, not only are we more stable than the other methods, but also we feel that our feature importances align well to the already known biological pattern in literature. You might be asking, what is the future of this research? Well, we are intending to apply this method not only to taxonomy, but there are many different ways to assess the difference between microbes, which might even be better than taxonomy. For instance, trophic networks which look not only at the similarity of microbes in the taxonomic tree, but more in the co-occurrence. So it's really a more correlation or co-occurrence driven similarity rather than only genetic differences. And not only are we intending to develop something like this, but also something like sequential transfer learning, where we first go from a model trained on ImageNet to a model which uses microbial data, but then we can then again transfer that model to a new target data set to actually go from something like ImageNet to a very large microbial corpus to a very small target corpus, where we have a model that is maximally trained on already known microbial interactions with, for instance, disease. Yeah, we feel that our model can also possibly be applied to different types of omics. So not only fecal metagenomics, which is the study of the human gut microbiome, but also possibly to metabolomics using metabolic pathways, which can be used as a similarity method or a distance method derivative, or also gene networks, which allows us to do epigenetics or transcriptomics analyses. So there's a lot of research ahead of us, and I look forward to hearing what your questions are, but not without thanking the organizing committee for this opportunity to talk to you all. Also, my supervisors, Max Nieuwdorp and Evgeni Levin, for all the help, as well as the bioinformatics department of the Amsterdam UMC. So thank you all for your attention, and